everybody for joining us.
Thank you, Lisa. My name is Marc Giroux. I'm the founder of Kurve Therapeutics. We have been pioneering bypassing the blood-brain barrier for a long time now. So I'm very glad to be here. I'm very glad to be on a panel with Anna and Tamara. You're going to find, I think, their presentations to be very illuminating. I'm going to do my best to do so as well. What we are not is a nebulizer. And that's what a lot of people seem to think, because we're creating droplets out of liquid. The problem with the pulmonary nebulizer is it's very, very good at getting drugs in the lungs. And if you put it in your nose, they're also going to get in your lungs. 67% of everything you put up your nose with a pulmonary nebulizer will end up in your lungs. The FDA does not allow that. So we have to make sure we stay out of the lungs, which we do. And I'll explain how we do that. But this little guy here has been around since 1887. So those of us of the right age remember watching movies where these people were using this for perfumes. Eventually, it went into medicine and nasal spray bottles. But from 1887 to now, they really haven't changed a lot. 90% of everything you put up your nose through a spray bottle is going to end up in your stomach. So really, until we came along, putting things in your nose was really just an alternate form of getting the drugs somewhere else, whether it's the lungs or into your stomach. We already did. We changed this. The company was founded on these principles. I have chronic sinusitis, allergic rhinitis, and asthma. So I was mostly just trying to change my quality of life. I was president of another company at the time. And we started doing this full-time when it became necessary. But just so everybody is clear, we started out being a topical and systemic company. We just wanted to treat the nose itself. Systemic means we want to get it into the blood for pain management, things like that. So we got good at that. That's where we started. Nose to brain came calling when they realized we could access point. The first hypothesis of doing something nose to brain was done in 1989, but it was never proven because there was never a way to get the drugs to the spot. You could prove it in an animal using a pipette, but in a human, it was virtually impossible until we came along. So I'm going to tell you how we did that. And the thing to remember is it's a physics problem. Okay, we're trying to get droplets to a certain spot in the anatomy, and we're going to have to fight the anatomy and biology to succeed at that. But we've succeeded at all of that. And we've succeeded fairly well. Alzheimer's is phase 3 ready. Mild cognitive impairment and diabetes is phase 3 ready. Parkinson's is phase 2 ready. All of these things are things we've done that have been published. We have a bunch of for-profit companies that we're dealing with that we're under NDA with, but we've been involved in over 50 clinical trials monitored by the FDA. So we are an ethical pharmaceutical and medical device company, and we have done a really good job working with the FDA. We are now, when it comes to nose to brain, we're pretty much a rubber stamp as far as they're concerned. Simply because they've evaluated us so many times. And essentially what they say now is, yes, if you're going to do nose to brain, we know Kurve works, you're ready to go. If it's a different device, they end up saying, well, prove to me you can do it. We had to go through the same process, so there's no reason why neither one of our competitors should have to do less. We started out with proteins and peptides, just trying to get something into the nasal cavity and then eventually into the brain. Our first one was insulin. Monoclonal antibodies, we were told, were too big. You'll never get that through the olfactory region. They said the same thing about polyclonals after we managed to do the monoclonal. We've also done combination products where we did 3 drugs at the same time into the brain to treat a brain tumor, and we were successful at that. So not only can we do combination products where you get 3 drugs in a single dose, but you could also do it from 3 separate reservoirs and get it into the brain as well. So there's a lot of capability here. So if you're a physician and you're looking at potentially treating a patient with more than one drug, which is very common, we can do that at the same time, or we can do that in series. So in parallel or in series, we can do that. The biggest thing we've delivered is a stem cell. The stem cells are huge. And we were told there's absolutely no way you're going to do that. Well, we did that too. So pretty much everybody in the world has just backed off and said, okay, Kurve can do this. They at least have proven it. For the regenerative world, we're really excited about all the things that we can do with these products. The Muse cells are brand new. We should get pricing on those pretty soon. I think Anna might speak about them in more detail. If she doesn't, we can certainly throw a lot of data at you with regard to that. But stem cells are the big player. Exosomes, Muse cells, and these rubella exosomes are secretomes of stem cells. Stem cells have tumorigenic properties, so to avoid that, you can use exosomes and Muse cells. Not only are they less expensive, they're easier to transport and easier to deliver, and it doesn't come with the same baggage that some stem cells do. But regardless, if it was what you wanted to deliver, we could and we have. So it's important to note that the technology platform is extremely versatile. We'll get into that in a minute. But the exosomes and Muse cells, we are really excited about all of these things that we're doing and all of these capabilities that they have. We're already working with both retired football players in the National Football League here in America, as well as active players. We're also talking to the actual teams and the team doctors and the directors of player personnel to get this officially into the team room as well. Those talks are actually going well, and the progress on that has been very high. In addition to that, the Department of Defense wants us to help with their soldiers who have concussions as well. So we're simply out there just trying to protect people's brains, help them heal. Where we're actually working with retired players to not only add cell growth, but to arrest the brain atrophy and maintain their quality of life. And there's a real opportunity to that with some of these products. So Kurve does not play in traditional medicine and regenerative medicine exclusively. We find them both to be redeeming, and we want to be the device of choice to do all of that. So we're not going to turn our backs on either one. So whatever you have that you're doing that you could use our help with, we're willing to work with you on it. In some of the cases for our PANS, children's, and moms, we have been supplying a lot of peripheral formulations, because once the PANS children start to do better on IgG, they end up needing potentially infection treatments, other things to enhance cognition. Some of them carry autism as well, and we have opportunities for that. And that's why the machine is, this large, because we can make this do anything we want. And it also allows us to have giant reservoirs if that is what a particular drug needs. Eventually, we're going to be selling these. Don't know if you can see the mist there. It's a little difficult. But these would come pre-filled. We would take them off, throw them away, and you just replace it so that your commitment to the device is only $1. As I was talking about, all of these things tend to have peripheral conditions, and these are some of the supplements that we can actually do as well. The bacteriophages are under prescription. A lot of anti-inflammatories are. Insulin is and is not. But a lot of these things are because the patients come with complications. Cleanses and detox can be helpful, and they can be harmful. My advice, having treated a bunch of these things at this point, is to only deal with the things you need to deal with. So if there's a sinus infection in your patient or your child, let's deal with that first. We cleared MRSA out of a particular patient who was under 10 in 2 weeks, and he'd had it for 2 years. There's a lot of things that go on that complicate some of these diagnoses, and we can help treat those as well. If we can take any liquid or any suspension and put it in the brain, then everybody has a lot of options if they come to work with us. So keep that in mind. It's not the first drug that you were given and put it in a device and you didn't get the results you were looking for, then we make adjustments. if you're going to deal with a spray bottle or pulmonary nebulizer, they're a one-shot deal. They will either can deliver it or they can't. And they certainly can't do it nose to brain. I think the important piece now is to manage your detox and your cleanses to the point where you're not overdoing it. It does some harm when it does some good. I'm not going to tell you what to do. I'm just merely giving you the benefit of our experience. How we do what we do is complicated, but for the use of the device, it's not. It's very intuitive, and we have Alzheimer's patients that live alone who can use it. So no matter what happens, everybody on this call will be able to use it. We've had 24 peer reviews. We have 300 patients in a study being done now in Southern California, 30 of which have completely resolved their symptoms. So we have Alzheimer's patients who are no longer demonstrating any symptoms at all. And one of the reasons for that is we get 3,000% more in the brain than an infusion does. Big Pharma doesn't like that. They make a lot of money from infusions. We actually cost them money because we do it much more efficiently. This is in regard to the IgG. So we get 3,000% more in the brain, so we can lower the dose, make it safer for the patient, especially for a child. The things that we had to figure out that I think people need to know why this works when nothing else does. And I've heard this a lot. I put the drug in a spray bottle and I got no benefit from it. Well, the reason is the anatomy doesn't let you. The black are the airways. We want to get to this olfactory region spot, which is right between your eyes. In addition to the anatomy stopping everything right here, the airflow pattern goes horizontally. So when I'm studying the physics of what actually happens in the nasal cavity, when I was just trying to get rid of my chronic sinusitis, these are the obstacles I had to overcome. So this is what a spray bottle deposition looks like. As you can see, it's not anywhere near the access point, which is right here. 90% is going to your stomach. We already covered that. So what I found we needed to do, and the first thing I tackled in the physics of this was droplet size control. Has to be bigger than a nebulizer, smaller than a spray bottle, but what's that sweet Goldilocks zone that the engineers could hit? And droplet size control became our first target. We can now do droplet size from 3 to 300 microns, one at a time. So if your drug, your patient needs 17 microns to get it in the brain, then that's what they get. If it's 22, 27, then that's what they get. Turbulent flow atomization rate and exit velocity tell us where it's going to go. So we manipulate those three things along with the droplet size, and that allows us to target. So for chronic sinusitis, this is a beautiful deposition pattern. Nothing in the lungs, nothing in the stomach, and we got all of this nasal cavity, and we hit the roof. But that's not beneficial for nose to brain. This is. We changed the turbulent flow atomization rate and exit velocity to put everything here as much as we can. And now you can see the drug is actually in the brain. I challenge any device out there and any drug company out there to produce an image like that. We've been able to do this for more than 10 years. There is no reason why this can't be done. This is why our device works with pretty much any liquid and any liquid characteristics. So a suspension, a high viscosity liquid, we can break it all down. As a really quick aside, because I know I'm running out of time, Estee Lauder wanted us to spray anti-aging on the skin. That stuff was almost toothpaste, but we had to create a droplet that would absorb faster than it evaporated, and we did that. And that allowed us to learn how to deal with high viscosity liquids. Insulin is not a high viscosity liquid, but we have great results. Here at month 18, you can see us still separating from placebo. This line is a competitor's line. It's also the placebo line. That competitor is now out of business. The last slide I have for you right now is this chief of dementias at the NIH here in the United States. And they recognize that we're not only getting the drugs into the brain, but we're actually changing the brain. We have biomarker changes that can be tracked. Amyloid plaque, we have completely eliminated from somebody's brain. But for the most part, IgG gets rid of misfolded proteins that your body's already doing. It's just falling behind. So I'd like to open it up for questions now. This is what we're capable of doing. I want everybody to understand that there is a difference behind what we do and what others can do, and why you can have confidence that we are doing this. It's actually true. We have the 22 peer reviews, but we have 50 evaluations by the FDA to tell us whether or not um, uh, we are safe and that we're effective. So I don't know of any better promotion than that. We're able to get the drugs to the place. People have verified that as a third party, and we have had tremendous success. If I can take another second, the PANS children have been really exciting to work with, and Kurve is looking to run a orphan study with PANS children, and we're going to be looking for a principal investigator to do so and actually apply with the FDA to do that. If you want to be involved in that, that's over a year away. We're just starting that process now. So it shouldn't affect your decision on whether you want to start now, because you can. We can sell these devices to anybody for anything as long as we have a doctor involved. So thank you for your time. It was my pleasure to be here. I'm happy to take some questions.
If I just may ask this question so that some of the parents will understand, it's based off of the Alzheimer's protocol. If you can discuss how what the immunoglobulins do to reduce neural inflammation, and then obviously just touch on the Alzheimer's regarding what the insulin does, what those two do to the brain and then the body and biomarkers.
Yeah, thank you, Lisa. That's a great question. Like a lot of things that are naturally occurring in your body, they have more than one job. So your body produces IgG and it handles getting misfolded proteins out of your brain. It's happening to you right now as we speak, and to me and everybody else on the planet. There are certain genetic predispositions, and there are certain patients who are just going to be more predisposed to getting this problem earlier in life, where the IgG is no longer keeping up. So what we're going to do is artificially enhance that. But what IgG does is it does attach to misfolded proteins. It's an antibody that will grab those and take them out. So we're clearing misfolded proteins and problems in the brain. It also does a really good job of neuroinflammation, and that happens very quickly. For an Alzheimer's patient, we don't even check in for 4 months. They've had the disease for a minimum 10 years, possibly 15 before they had a symptom. So the path back is longer. It's different with PANS. With the IgG, within 48 hours in some cases, the kid is totally different. And we've gotten a lot of them back in school, which we're really proud of. The insulin does a different job in the brain, in that it really does a good job with promoting verbal fluency, that's searching for a name or a word. It also does good with short-term memory and attention span. What it doesn't do is actually attack the root cause of the disease, the pathology of the disease. The IgG does, but it doesn't do it in every part of the brain. So we need to be able to target more in the brain and move some things around. And that's where we think the mu cells and exosomes are going to do a good job. In addition to the IgG and the insulin, we have a handful of patients who are using both, maybe 25 or so, and they're doing quite well. But the IgG got 2 Lewy body dementia patients out of a wheelchair. They couldn't walk, couldn't talk. They were visually hallucinating. They walk and talk now. They walk and talk to me. I can get them on a Zoom and they'll walk over and say hi. They recognize their family members. There are paths back from these conditions that people don't realize, and they're actually happening now. But we're not allowed to publish things that, we haven't formally completed all of the trials on. So we can do them stage by stage. We're not allowed to market or advertise. This is the best we can do, is to simply tell you what our results are, but I can't run a commercial on it. So if you want more detail and you need more data, we have 15 years worth of data. I could do a 200 slides on just the data. If you're a physician out there or an interested patient, ask me for it and I'm happy to share it.
Okay, we have this question. How do you ensure that no bacteria or other elements get in the device and cross the blood-brain barrier?
Can't. There is no way to there's no way to manage that. I can only tell you that in 15 years and thousands of patients, it has not happened even once. Um,
And then yeah, explain like how one would clean the device to guarantee this, how others are keeping the device clean to guarantee that.
Yeah, there's nothing different about our device as far as that goes from a pulmonary nebulizer. And that's kind of where I started in my journey on this. Because I have asthma, I owned one. I actually owned a bunch. I kept buying them as they got smaller. Um, so the routine, if you have asthma and you have an asthma attack, is get your nebulizer out, you use it, and then you clean everything in your sink. You know, soap and water, hot water. Ours are all autoclavable. It's not something every patient's going to do, but it can be sterilized with an autoclave or with isopropyl alcohol. But for the most part, patients just clean it out in their sink, run it through. Most of the things we deliver are biologics that will attract mold. So you got to be diligent about cleaning it. But mold can be seen. So you just got to pay attention and know what's going on. But we have not had a single event in the brain yet. We also haven't had any brain bleeds, which our competition got as much as 35% of the time. That's more than 1 in 3 patients are going to have a brain bleed. We've had 0. So as far as getting things in your brain, if your brain can let things through the blood-brain barrier that cause problems, it doesn't happen often. We have never had it, but I'm not saying it's impossible. But there's also no way for us to protect against it. It's just a more efficient way of doing it. Benefits outweigh the risk, especially since we haven't seen it yet.
Okay, and then Alberto does have a question about organizing a meeting, which we're more than happy to for Pan's children outside the United States. And so just for anyone, contact me or the info at Kurve Therapeutics, and we'll be able to coordinate a more in-depth one-on-one. And yes, we are actively looking for, we're in the informal stage now of collecting data for the pediatric PAN study. However, as Mark mentioned, we want to educate. Our goal is to educate as many practitioners as we can and find practitioners outside the United States as well who are willing to treat, as well as that's why Dr. Anna's on board, and we are doing a a treatment with regarding medical tourism and PANS Panda / long COVID. So there's a few ideas. We'll contact us, think tank or Kurve Therapeutics, and we will figure this out. So I just sent a yes to that. And then what kind of responders to non-responder ratio are you seeing in your trial participants?
I do not understand the question.
So I would assume, like, let's start with the Alzheimer's, and then we can work over to Parkinson's, because that's another fabulous protocol with glowing, remarkable results. So in Alzheimer's, how many were non-responders? In how many, just a generalization, in Parkinson's were non-responders? And then
I see. They didn't get a benefit from the
yes.
from the actual treatment. Okay, I was thinking they maybe were looking at inclusion exclusion criteria. Okay, the in the insulin trial, this is all part of the public domain, so you can go verify what I'm saying. 3 out of 4 patients in the insulin trial regained cognitive abilities they'd lost, so they actually tested higher coming out. Than when they went in. We have in the 1,000 patients that just are involved in insulin, we had 4 that declared they got no benefit and then stopped using it and did not return. We had a whole slew of people who said, we're not getting anything, so we're going to stop, and the patient nosedived. So what that meant was we had arrested the disease from progressing, or it significantly slowed it so that they couldn't tell the difference. And then once they got off of the therapy, they started to regress again. So that's a good testimony to how the device works. So that out of the 3 out of 4, that 4th one just got arrested. So we didn't see any that didn't get any benefit other than those 4. And in the IgG, it's been completely 100%. They've gotten something out of it. So they actually feel sharper and they're okay with it. They're not necessarily having all their symptoms resolved, but there's been a definite improvement in slowing in the development. Parkinson's, we had a couple of patients who are also, we have one in particular who was being fitted for a wheelchair because he kept falling down. He plays tennis now. He also went back to work. They just said he's taking a break from Parkinson's. So it's there has not been a ton of people who didn't get a benefit, mostly because something actually gets in the brain and it does something. With the IVs and the infusions, when we measured it, it was 0.1% that could be found in the cerebral spinal fluid. We got 30 times more than that in the brain. It's 3,000%. That's a lot. So we can do a lower dose, make it safer, still get better efficacy than an infusion. Saves a significant amount of money, which is why Big Pharma does not like us. You know, if an infusion is $10,000 a month, we cost $350 a month. You know, you can see why they don't want to make less money.
Then another question is, if a drug is compounded with a suspension, would that be able to be used in the device? And so then I guess this is also a time to make note that you're such a fabulous company that you tell us, explain your process about pretty much working with any medication, any supplements, any regenerative medicine product.
Yeah, it's a good question. We've done suspensions. We've been very successful with them, especially when we end up with, you know, something in a dry powder that has to be reconstituted. But we actually had a solution and a suspension in a single reservoir. And you can go look this up. It's called Ciprodex. It's a corticosteroid, which is an anti-inflammatory, and which is the dex part. And it was ciprofloxacin as the antibiotic. And that's generally an eardrop, but they wanted to put it in the nasal cavity. So we did a solution and a suspension at the same time. So this is not a problem. The action for the patient is either to mix it from the powder into a suspension at the time of dosing, or like any other anti-inflammatory, you just got to shake it first and then you deliver it. It's really quite simple and not complicated. There is a milling issue with the size of the particle that is being milled down to for the suspension, and we should talk before you go to the compounding pharmacy about what that needs to be.
It is supposed to be clear, baby. I will hand you.